All right, so as you go over this then, so for instance, B, you should have that the only one that occurred that was different for B is and 3. So you should have a B here, a change in trait B here, so B, and a trait change here. And then for C, you have 1 and 3, both had a change in there. So you'd have 1 and 3 here, so on C here, so you'd have 1 and 3. 1 and 3 here are off the same branch here, so therefore you would only need to have it change one change here to change both of these here, and then 1 and 3 here. And then for D, the only one that changed was species 1, so you'd have a D here, so I'll D here and a D here. And then lastly, E, the only one that changed was species 3, so you'd have E here, and E here. All right, so that's what it looks like. So then what you do with this, this is called, we're looking at parsimony, which is, says that the tree that is most likely would have the least changes or the least number of events to make that tree happen. So which one of these has the least likely events? Leo. Number 2 here. So I have here, we have 6 changes here, 5 changes here, and 6 changes here. So, so looking at this morphological data, tree number 2 is the best fit here. But which one was the best fit for the DNA data? Remember? Yeah, number 1 was the best fit for the DNA data. So therefore, the morphological data and the DNA data don't agree here. And this, the last question, so the first question along there underneath there asked you which of these phylogenies is most parsimonious, and you should have, for this activity here, it was number 2, but when it says how does the results compare to the result given in figure 26.15, that's the one that we did in class together with the DNA data. For that one, the first one was the most parsimonious, they had the least changes. So then the last question asked you which of these phylogenies, which tree here, the one that we just looked at with the morphological data or the one with the DNA data, is more correct. Do you guys have an opinion on that? What you think would be the most correct if you're looking at both of those more physical features versus the DNA data? Lucas? Probably DNA. What makes you say that?
Because it's actually happening and just not just based on characteristics, which can look similar but can be caused by differences in DNA. So
Yeah.
it's like more specific.
Yeah, absolutely, absolutely. And so, so the DNA one should be, and if we have DNA evidence, that's the one that we would use. We'd look at all of the evidence and see if they kind of coincide as well. One thing about this particular DNA evidence that I would say is that in real life, we probably wouldn't look at just 4 bases, because 4 bases in DNA, not very long. A strand of DNA can have millions of bases long, and so therefore you probably look at a longer strand and look at it. They just chose 4 for simplistic sake to show you here, but realistically they would use more data. All right, on that. All right, very good. All right, so let's move on here in the notes then. So this is why it says phylogenetic trees as hypotheses. So this is we have a hypothesis based upon evolutionary relationships based upon the data that we have at the time. That data can change, and therefore evolutionary trees can change. So the best hypothesis for phylogenetic trees fit the most data. So that's morphological data, molecular like DNA, fossil data. And this bracketing allows us to predict features of an ancestor from features of its descendants. So if we look at have this bracket, we have the descendants, and all the descendants have certain characteristics, then we can say, okay, then that probably came from a common ancestor and look at the ancestors of that. So for example, the bracketing allows us to infer characteristics of dinosaurs based upon their descendants. So what characteristics might they have had based upon organisms that they are related to. Now this doesn't prove, infer characteristics doesn't prove that those characteristics are there. It just allows you to say, here are these descendants, they may have had this. It's not until you find concrete evidence that we can say it for sure happened. So what would be concrete evidence, like fossil evidence, and so on. So, an example, birds and crocodiles share several features. They share a 4-chambered heart, song, so they have a certain song that they quote-unquote sing, although the crocodile song is not as pretty as the bird song, all right? But, and then the nest building and brooding. Brooding is sitting on the eggs, all right, in the nest. So birds and crocodiles have these features. These are descendants, and so these from a common ancestor. So these characteristics that they have in common most likely evolved in a common ancestor, and then were shared by all the descendants of that common ancestor, and the descendants of that common ancestor also would be dinosaurs. So the question is that dinosaurs, since birds and crocodiles share these 4 characteristics, and they have come from a common ancestor in which also dinosaurs have come from that same common ancestor, do dinosaurs have these things, all right? And so that's what we're talking about inferring, but it doesn't prove. And so, so when we find things that prove that, it supports that inference. So the fossil record supports nest building and brooding in dinosaurs. They found in certain species of dinosaurs fossils that show nesting and brooding. And so this is a picture of one particular fossil. The picture is on the left, so this is the actual fossil. Here are eggs, right here, some more eggs here. And then these are fossilized bones of the front and behind limbs of the larger parent dinosaur. And so from this, in studying this is an artist's reconstruction of what this parent may have been doing with these eggs before they died. And so then this is a showing nesting and sitting on top of their eggs, which is the brooding part. All right, and so it's not until you find evidence that you can actually prove. When we, like I said, going back to here, birds and crocodiles do this, and they're related to dinosaurs, so we can infer that dinosaurs does that, but not until we find evidence like this. All right. All right, so this is a linear tree to show common ancestry of, we just talked about the crocodiles and the birds, all right, so having a common ancestor, but we also see the dinosaurs. And so as we find new information, it changes the shape of our trees. So we can revise our understanding of phylogeny and how organisms are related, and what this tree of life looks like. So recently we've gained insight into the very deepest branches through molecular systematics. This is analyzing DNA and molecules. 80 years ago, we weren't able to do that. And so everything was based on just morphological features, fossil features, and so on. And now we can look at and actually compare DNA, which gives us a new window into relatedness between organisms. And so we've changed some things. So we've gone when kind of categorizing all living things on the earth, we've gone from 2 kingdoms. So at one time, early taxonomists classified all species as either plants or animals. So they put them into these 2 categories. And then as technology came about, we realized, okay, we have a lot of unicellular organisms that we weren't able to see until there was a microscope to be able to see them. That there's all these unicellular organisms swimming around in the lake that we're swimming in, and they don't really fit into a plant and an animal category. And so then we said, oh, we have to revise everything, and we kind of revised it to include those other organisms organisms, and so on and so forth. So our kind of tree of life and how we have drawn this has really changed over time. So then later, 5 kingdoms were recognized. The first kingdom was Monera. That was all bacteria, all prokaryotic cells. Protista were unicellular eukaryotic cells, and then we had the plant, fungus, and animal. And so this is what I learned in high school, all right, is the 5 kingdoms. All right, and so then, since then, that has changed. And so now we have that 3-domain system that we talked about 2 weeks ago. The kingdom are the domain bacteria, archaea, and eukarya. Remember, these 2 are prokaryotic organisms, and everything made out of eukaryotic cells falls in that domain.
So if they were to add, viruses to this tree, would this be under a domain or a kingdom?
I would believe that it would be because it's not like anything else,
Okay.
which is
So it might be
why it's not on
added.
here. All right.
So one day
So-
we're teaching a biocustom. Back in
Well,
my day,
that's right. And I'm up here with my walker.
walk around the walker.
Walker with the walker, that's right. Back when I remember teaching about, this the 3-domain. Yes, absolutely. And I'll say, I had a student named Liam. All right. And how do they know this? What they decided, this Monera kingdom, they put all the unicellular prokaryotic cells in one kingdom, and then upon further analysis, figured out, whoa, these bacteria, that we clumped all in one kingdom, actually have really big differences between them. And so we can't put them all in one category, we need to separate them out. And so they were different enough that they put the bacteria and the archaea, the prokaryotic cells, some of them in here and some in here, into their own separate domains, all right? And that is when we were able to do that by looking at DNA and analyzing it. So this is a picture of a current tree. Here's the common ancestor for all life here. Here's the circles represent the 3 domains. So here's the bacteria and the archaea. And then we have everything that's made out of eukaryotic cells here. So we would be here in the animals here. That's where we would fit in here. Notice here, a common ancestor here, and we have 2 branches coming off of here. So are we more related to the domain archaea or the domain bacteria based upon this tree of life? Are we more closely related to the domain archaea or the domain bacteria? And it is archaea. Here we are, all right? So here we are in the domain eukarya. If we go back here in this branch here, our most recent common ancestor here with the archaea is here versus here for the bacteria. All right, so let's look at this. So to even be put on the same tree of life, all living organisms, no matter what domain they're part of, have to have certain characteristics. So they have DNA and RNA as carriers of genetic information. All living things have to go through transcription, translation, and replication, which means that, like, replicating the DNA. The features of the genetic code are shared by all modern living systems. So what does that mean? DNA all organisms has the sugar phosphate backbone, and you have your bases, A's, T's, C's, and G's that come out and bond together. And every organism on Earth, the DNA has the exact same structure that way. It has all the same 4 bases, just the order of those 4 bases and what proteins are made that are different between living things. And then certain metabolic pathways are conserved. So for instance, being able to break down glucose to make ATP. That's something that's in common for every single living organism and they're able to do. And so, so those are some of the things that all living things have in common. Let's look at the domain eukarya. For eukaryotes have certain things in common to put them in their own domain. So we have in our cells a cytoskeleton that's made out of microtubules and microfilaments. We have structural proteins that help with cell movement, help with the shape. membrane-bound organelles like the mitochondria, linear chromosomes, unlike in prokaryotic cells that are circular chromosomes, and then the endomembrane system, which kind of goes with the membrane-bound organelles, that's the rough ER and the Golgi apparatus and all the different transport vesicles and so on. And so, looking at now our most recent tree of life, as we looked at earlier in the kind of branching tree, that eukaryotes and archaea are more closely related to each other than to the bacteria in that particular domain. And how they've analyzed this is, says the tree of life is based largely on rRNA genes, as these have evolved slowly. rRNA stands for what? Ribosomal RNA. Remember that in our DNA, let's just do a quick little review about DNA. We've learned that genes can code for making proteins, but how they do that is they make an mRNA, and that mRNA is read to make proteins. But not every part of your DNA actually codes for making mRNA. There are two other kinds of RNA. There's rRNA and tRNA. And so therefore, right here, this part of the DNA might make rRNA. This has nothing to do with making proteins. It has everything to do with making ribosomes. So this actually goes to the physical construction of ribosomes. And then other parts of your DNA code for making tRNA. The tRNA don't literally make proteins, but what they do is they help with that. What do? tRNA picks up what? Amino acids, right? And bring the amino acids and so on. So this is what we're talking about here. Looking at rRNA genes, so it's not genes that necessarily make proteins, but rRNA. And then another thing that makes a little bit different in studying the tree of life is that there's been interchanges of genes between organisms in different domains. And so what this means is they call this horizontal gene transfer, the movement of genes from one genome to another. So this is different. I want to point out here that when we pass our genes on to our kids, that's called vertical gene transfer because you're passing it on to the next generation. You're passing your traits on. Horizontal gene transfer is between two organisms on Earth switching genes. So it's like one organism giving genes to another organism. Can anybody think of a situation we've talked about in this class where organisms can get genes from another organism? Do this horizontal gene transfer. Okay, so interbreeding between similar species, so like the lion and tiger that we talked about, absolutely. The interbreeding, though, that's more passing your genes on to the next generation. Horizontal is more one organism that's alive today gets new genes from another organism. Got one? What was that? Viruses, all right? So viruses, what they do is they inject genetic material into their host cell. And so viruses can inject their own DNA into the host cell, but sometimes viruses make a mistake and they can infect one person or one organism's cells. And then when they make new viruses, they accidentally put some of that DNA of that organism into the viral virus. And when the virus goes and infects another individual, it can inject the other individual's previous DNA into it. So that can happen. So viruses can do this. I had some other hands up. Everybody here was going to say virus? Yeah.
Just wondering, horizontal. So yeah, we don't have bacteria use that with the horizontal gene transfer when they make that weird link between them.
Mm-hmm. Yes. So when they sometimes called the mating bridge is the sex pili, where they this right here between the. Is that what you mean? Kind of where they bacteria, can swap DNA. Anybody know what that's called? Conjugation. All right, so this is conjugation in bacteria. All right, and so that's one way that they can do that. Another way is the bacteria can take in, remember, plasmids from outside sources as well. So this transformation, we did that with in the lab as well. It's another example. All right, so horizontal gene transfer occurs by exchange of transposable elements. This is transposable elements. Another way is called jumping genes. So genes can jump from one chromosome to another. Plasmids, that's like in the transformation that we did. Viral infection, and fusing of organisms. So if organisms fuse together, so this would be like, it's kind of like conjugation here where they temporarily fuse and therefore can exchange DNA. And so when this happens, it complicates efforts to build a tree of life, because if we're looking at particular traits and organisms are swapping genes, that makes it a little bit hard to follow through time. And this is the another way to look at the current tree of life here, where here's the common ancestry of the domain bacteria coming off first, and then you have the eukarya and the archaea coming off together. All right? And that is chapter 25. All right, we're going to move on to chapter 26.
Fill out my notes for me.
Roughly 8% of human DNA is viruses. Wouldn't that be like, hey, they introduced our genome?
Interesting. That's not high percentage.
Yeah, 8%.
All right, so what this last chapter with us is about is looking at the macro part of evolution, like in terms of when things happened and looking at the history of the Earth. And so the first part of this, the first section, is about the early Earth and how we get organic molecules in cells. All right, so chemical and physical processes on early Earth may have produced very simple cells through a series of stages. So think about what cells are made of. First of all, we have to have small organic molecules. So it says here, abiotic synthesis of small organic molecules. Abiotic means without life, it's not within life. So organic molecules made outside of a living thing. So an example of an organic molecule that I'm talking about here would be like amino acids. So example, amino acid would be an example of one type of organic molecule. Then the organic molecules have to join together into polymers. So if going with my amino acid example, if we were to make a polymer from this, what would the polymer be? A polypeptide, which then could eventually form into a finished protein. Then we have to have the molecules packaged into, it says, a protobient. This is a precursor of a cell, and I'll talk more about this in just a second. So it's spherical. And then origin of self-replicating molecules. Self-replicating molecule. What's a self-replicating molecule inside cells that are really important? That can self-replicate. When it has it's a molecule. DNA is an example of that. All right, so all these things things need to kind of happen in order to get life. So we're going to talk about each one of these and go through it. So the first one is the synthesis of organic compounds. So little history here. Earth is roughly 4.6 billion years old. Its early atmosphere is very different than today's atmosphere. So early atmosphere contained water vapor and chemicals released by volcanic eruptions. So it's very volatile, it was very warm, a lot of volcanic eruptions putting certain gases in the air. When it says chemicals released by volcanic eruptions, what it did not have is oxygen, so no O2. So some experiments have been done to simulate the early Earth atmosphere and showing that you can produce organic molecules from these inorganic molecules that were most likely found there. It says, but such an atmosphere on early Earth is unlikely. We'll talk about that in just a second here. They think it's more likely that the organic molecules were formed not in the atmosphere, but either by hydrothermal vents in the water or due to volcanic eruptions in the water, due to volcanic activity in the water. Let me show you this experiment that was done within the laboratory by two scientists by the name of, their names were Miller and Urey. This is the Miller-Urey experiment. So what we had, they tried to simulate what they thought to be the contents of the early Earth's atmosphere. So this is a closed system, with the exception of they have a little stopper that they can unplug and get water. This blue stuff is water here from the system. So let's look at here. Notice here there's arrows going this way, so everything flowed kind of in the clockwise direction. So let's look at what's in here. So they have water here with a heat source, so it was very warm, and you get the water evaporating and get water vapor. So here's your water vapor. Enters into this chamber. They put gases that are thought to be in the atmosphere due to the volcanic explosion on the Earth. And so that's these guys here. On these electrodes here, simulating, what do you think electrodes are simulating there? Electricity, under volcanic eruptions.
Would it be radiation from like the sun?
It could be like.
Since we had no atmosphere left.
That's right. It could be that. Also maybe lightning as well, but more electrical current there. All right. So they're trying to simulate what the Earth atmosphere would be like, and so that they allow this water vapor with these gases to cycle through here. And notice here that as it comes down here, what I want to point out here is this is all connected. So whatever's in here stays in this tube. But what they have on the outside is kind of like a tube that's kind of a donut tube around it. So if I were to look at maybe the cross section of this, so this right here, if I were to take the cross section of this would be what's inside of here. And then around it, you'd have cold water flowing around it. So notice here, cold water coming into this tube around this inner tube. And this cold water then comes out, and so the new cold water constantly comes in. What that does is cool off whatever's inside here. So all this water vapor inside here cools off. What happens with water vapor when it cools off? It condenses. So the kinetic energy, it loses kinetic energy. The water molecules get closer together, and they form liquid water. So that's what they're showing you here is this droplets forming liquid water. And then what they did is took samples from this water coming from here. And when they tested those samples, they found that in the lab, these two guys here found that the water contained organic molecules. So an example of what they found was they found certain amino acids. Okay, so that would be an example of what they found. And so this was a big experiment. Why? Because it showed that organic molecules could be made from molecules that are that could have been on early Earth. All right, so that's the significance of this. So I'm going to put here that, so the significance, organic molecules made in lab with early Earth conditions. That's the significance of this picture and what they did. They call that abiotic synthesis because it was made outside of a living thing. All right. So, as I said earlier, that instead of forming actually in the atmosphere, that they think that maybe the first organic compounds may have been synthesized near submerged volcanoes and deep sea vents, but you still have the similar gases and things like that produced. And so this is a underwater sea vent. All right. We also have, notice here, extraterrestrial sources of organic compounds. So coming from space, all right? This does not mean aliens brought organic compounds to the Earth, all right? So let me explain why this is in here. So some organic compounds, notice it says some. It's not all. So this is not a hypothesis where all of our organic compounds came from somewhere else, all right? So some compounds from which the first life on Earth arose may have come from space, and this is the reason why. Meteorites, they found meteorites that have carbon compounds that have been found in them, that have landed on Earth. So because these meteorites have carbon compounds, that may have been some of the source of original carbon molecules on Earth. All right, and so that's what the extraterrestrial refers to.
So there's a theory called the pan-terrestrial theory that says that there was once life on Mars, and because of running water and whatnot, and Mars got hit by like a giant asteroid which wiped out all life on it, and then one of the fragments of this Martian rock then became an asteroid and hit Earth. Some of the bacteria live, because we know that bacteria can live in space for a long time due to the shells they create, and that's how there's life on Earth. That this happened. Just saying.
Okay. All right. All right. Interesting.
So we're all Martians, basically.
We're all Martians. Okay. According to that theory.
According to that.
All right. And kind of going with that same idea, as our technology becomes better, we have been looking for clues for life out of this planet for a long time, and so the possibility that life is not restricted to Earth is becoming more accessible as we get new technology to be able to look at other either planets in our solar system or maybe one day outside of our solar system, and so on. Because they found other stars that have planets revolving around them as well, and so, who knows. All right. Abiotic synthesis, now once we get the organic molecules, now we have to put them together to form polymers. So small organic molecules polymerize when they are concentrated on hot sand, clay, or rock. So they've actually found that, so we can make organic molecules like amino acids in the lab. But then how do the amino acids hook together to start making polypeptides to make proteins? So they've actually taken solutions of amino acids and dropped them onto hot sand, clay, and rock in the laboratory, and that the heat catalyzes the reaction of hooking the amino acids together, and the amino acids actually do polymerize. So that shows abiotic synthesis of polymers. Then we have to have the cell. This is the protobiont. These are aggregates of abiotically produced, so outside of the living thing produced, molecules surrounded by a membrane or membrane-like structure. So experiments demonstrate that they could have formed spontaneously from abiotically produced organic compounds. For example, small membrane-bound droplets called liposomes can form when lipids or other organic molecules are added to water. Now, I've talked a little bit about liposomes before, where when we talked about phospholipids, remember phospholipids have a head and a tail. The head is polar, the tails are nonpolar, and when you put them into water, they will arrange themselves into a bilayer and into a sphere. And so that's what will happen. And so this is kind of like what this is talking about here, this protobiont. And so if it's just lipids and water, you'd have water inside and outside of this kind of cell precursor, because it's not really a cell. And then if you add any other molecules in the water, so I'm just going to put here like molecule A, B, and C, that can be inside this solution as well, and those same molecules can be outside here. So you have a precursor kind of to a cell with certain molecules that randomly, when these phospholipids formed the shell, these molecules randomly got put into the inside of here, and then these guys are on the outside. So this would kind of be like the precursor to the cytoplasm and extracellular fluid, which is what this picture here is showing you, that these are actual pictures of these forming. So each one of these little circles are a different kind of protobiont or precursor to the cell. And what they've also found is that if they add molecules to the water and allow this to happen in the laboratory, I just put randomly molecule A, B, and C in here. But this is showing you actual real molecules, so they added some enzymes into the water before they allowed this to before it formed and so on. So these guys these molecules randomly get put into the cell, and these enzymes can actually, if the substrates are there, reactions can happen. So this enzyme binds with glucose and this glucose phosphate and helps to make starch and so on and so forth. And so reactions can actually occur if you have the actual chemicals in it. And so that's an example of a precursor to a cell. And then the last thing that a cell would need is genetic material. And they think that RNA was probably the first genetic material. And this is the reason why is because of some things that RNA can do. So first genetic material was probably RNA, not DNA. RNA molecules called ribozymes have been found to catalyze many different reactions. So ribozymes, ribo coming from RNA, because this is RNA molecules, and zymes kind of acting like an enzyme. What do enzymes do? They catalyze, all right? So RNA molecules are kind of like thought to be like the first kind of enzymes. So what they can do is self-splicing. So what does splicing mean? Remember we talked about splicing when we talked about cutting out introns, all right? Absolutely. They can make complementary copies of short RNA, so they can copy themselves or they can copy other short pieces of RNA. So therefore RNA is a good hypothesis is about why it might have been the first genetic material. Alrighty, I'll stop here. All right.